Okay, so thank you so much, guys, for coming. Today's lecture, we're going to study eye vision. We're pretty much going to talk about all the structures of the eye, the accessory organs, such as the eyelids, the lacrimal apparatus, and then we're going to talk a little bit about the extrinsic eye muscles, and then finish off with the receptors in the eye, the photoreceptors, the cones and the rods. Okay? All right, so since the LensSmart module was very easy for this one, there was not any questions that people had troubles with, but we got a fair bit to cover, though, in today's lecture. So we all know about the eyeball with the eye and the optic nerve, of course. Optic nerve is optic nerve number, cranial nerve number 2, right? Okay, so the eyelids, the eyebrows, the ones on the top, the eyelashes, lacrimal apparatus, extrinsic eye muscles, all of those are accessory organs that assist with the, sense of vision. And then, of course, we got the sensory neurons for it, which run along the optic nerve. All right, so this is just a picture of a kind of showing the position of the eyeball, and the iris is pretty much the curtain of the eye that regulates the amount of light entering the eye. And the pupil is that hole in that curtain, all right? And the iris will have two kinds of muscles, radial muscles that radiate outside, and then circular muscles. And we'll talk about those kind of muscles. They're smooth muscles, single unit smooth muscles that are controlled by the oculomotor, okay? And we will talk about conditions in dim and in light, and we'll talk about those two muscles and how they respond. Of course, the eyeball there, we got the sclera, which is the outer fibrous layer. It's in the fibrous tunic of the eye. It's got 3 tunics, the eye, and we'll go through each one of them: the fibrous, the nervous, and the vascular. All right. So, and then of course the lens, which refracts light or focuses light. Refraction is bending light to a focal point at the back of the eye and the retina. All right, and then so we talk about all those structures. The eyebrows, of course. What do you think the function of the eyebrows is? Yeah, especially sweat. Especially sweat, because sweat runs down your brow. If it wasn't for the eyebrows, all the sweat would run in there, and you know how sweat is salty. It would really clog your vision. It's very helpful when you're jogging, so it prevents perspiration from getting into your eyes. And then, of course, the eyelids. Okay, they got 5 tissues. The eyelids do the thin layer of skin on the top, and then like loose connective tissue, which is like fat, and then muscle that opens and closes the eyelids, and then the dense connective tissue, which is also called the tarsal plate, where the tarsal gland is located. And then the 5th one is the mucous membrane, which is the conjunctiva. All right, and I'll show you a picture of that in a little bit. All right, so you got those 5 tissues that protect the eye from foreign objects. All right, and also lubricate the eye by spreading tears throughout. All right, and we've got this meibomian gland, which is also called the tarsal gland. All right, and it produces an oily substance that kind of stops sweat from running out. You see, if you didn't have that sort of oily substance that kind of covers the lining or the edges of the eyelids, the tears that are lubricating the eyeball would kind of sort of, you'd be like crying all the time. You see, because the tears will just come out. So that gland does that. And then the ciliary glands, which are, located between the hair follicles of the eyelashes, okay, they produce, these are sebaceous glands, so they produce sebum. And sebum is kind of like a semi-solid oily thing, just like the sebaceous glands associated with sweat glands. All right, and the ciliary glands will produce an oily substance, and they're between the lashes strategically, so that they can make them nice and soft It's just like when you put oil on your hair. Otherwise, they will be so dry and so forth. So it's the oil that will go onto the eyelashes to keep them nice and soft, so they can keep particulate matter from entering your eye. All right? So, and then eyelashes, of course, you know, they're on the margins of the eyelids, and apart from, you know, protecting particulate matter from entering the eye, they also cause the reflex in, you know, for blinking. What's that?
Something gets too close to your eyelashes, you blink.
Yeah, you blink too. That's because the eyelashes are the ones that initiate the reflex for blinking, right? The eyelashes are the ones that initiate the reflex for blinking. So that's also very important. All right? Because the blinking is to do what? What is the purpose of blinking? Why do we blink? Well, to clean it, yes. But what are you really doing? What substance are you spreading throughout? Tears. Yeah. And tears do clean, because tears have also lysozyme, which are enzymes that will break down, you know, chemicals, foreign chemicals. It's kind of like the immune system of the eye. So the first line of defense for the eye. Because anything that's open to the external environment, you know, bacteria and so forth can get in. And then, of course, the conjunctiva, which is a mucous membrane that lines part of the eyeball and all of the eyelid on the inside. So that's why, you know, it covers part of the sclera, which is the white, very fibrous, tough part. And when you've got conjunctivitis, what do you see? The white part of your eye is red. That's the part that's covered by the conjunctiva. All right? And then if you open up your eyelids like that, then you see also it's red, because the conjunctiva runs along there too. Yeah, pink eye would be the conjunctivitis. Yeah, inflammation of the conjunctiva. All right? So covers the inner eyelid and also the anterior part of the eye, which is the sclera. The sclera is the white part of your eye. All right? So the corner here, that's where you have the meibomian glands that produce that sebum, you know, that kind of lines the outline of the right here, the caruncle. It's called the caruncle here. This is the medial angle, and this is the lateral angle. All right? That's why in the morning when you wake up, what do you wake up with? Gunk on your inner corners of your eye, you know, because all that sebum that's produced kind of settles there, you know. And if there's an infection, usually you produce a little bit more of it, and your eyes wake up really gunky. Yeah, that's not good. But yeah, it happens. So there we go. Oh, crap, did I go fast? So this, you remember the layers that I was saying of the eyelids, you know, so we said the first one is the skin on the outside, and then you see the skin right here, and then the connective tissue here, loose connective tissue, second layer, and then this muscle here, which is the orbicularis oculi, and also the superior palpebrae superioris, levator, palpebrae superioris. It opens the eyelids, all right? That's the third layer. And then, of course, the dense connective tissue, which forms the tarsal plate right here. It's on both sides, I mean on both top and bottom. And then, of course, the conjunctiva is the final one, all right? So those are the 5 you know, layers of the, so if I were to ask about them, you'd mention the skin, you'd mention the loose connective tissue, you'd mention the muscle that close and, you know, open the eyelids. You mentioned the dense connective tissue, or the tarsal plate, if you like, and then, because in it is the tarsal gland that produces, or meibomian gland. The tarsal gland is also the meibomian gland that produces sebum. And the conjunctiva, of course, is the mucous membrane of the eye, which is number 5. Mucous membrane. All right? Yeah, that's the order they go. Yeah. You know, the skin on the outside, loose connective tissue immediately. This is like subcutaneous fat, really. And then, of course, the muscle, the dense connective tissue, which is the tarsal gland. All right? Where the tarsal gland is located. All right, and then of course the conjunctiva on the inside. All right, and you could give functions of most of those tissues. Of course, skin on the outside and the muscle, what it does, and then the tarsal plate, of course, houses that. The tarsal gland, or the meibomian gland. All right, very good. So, more accessory structures. We've got a lot of accessory structures. Then we've got to go through the retina with all the cones and some of the specialized cells that modify the, the signal that goes through the optic nerve, which is sensory, right? So, lacrimal apparatus consists of the lacrimal gland on the top lateral corner of the eye, and then the ductwork that take the tears eventually to the nose, right? And so those are the canaliculi, and then the final one is the nasolacrimal duct, and I'll show you a picture of the of how the tears are produced from the top here, from the gland, through the canaliculi. They get into the eye. The eyelids spread the tears throughout, lubricate the eye, and then if you, ladies, when you're doing makeup in the morning and you're putting that line that surrounds your eyes, if you go like this, you will see a little hole in there on the corner of your eye. The guys who don't do the lining thingy, some guys do though, but you can go home and you know what I'm saying? You can go home and look, and you can see it. You can see a little hole actually. You can see it, and there's another one on the top. So the tears go through there, through the canaliculi that connect to the nasolacrimal duct.
So if you cry really hard, you get the
Oh, that's exactly what I was going to say. Or most of the time, if people are choking up tears and trying to prevent crying, they will still be crying because the tears will be coming and they get a snotty nose. You get a snotty nose all the time, right? If you notice when you're crying, you get a snotty nose and you'll be blowing. Those will be the tears going down there. Yeah. It's like an overflow thing, you know what I'm saying?
Some people can taste their eye drops.
Mm-hmm.
They dilate my eyes and I can taste eye drops.
Yeah, you can, because it can go through the posterior nares, because the posterior nares are connected to your throat. If you watched America Got Talent, you see some guys, they drink milk and then they squirt it through those canaliculi. Yeah, there was a guy who did it last time. Yeah, that's kind of bad, man. Because they need whatever is in that milk and stuff, you're putting it through the ductwork. You know what I'm saying? Sometimes milk is going bad a little bit. So for that reason, the tears have an enzyme called lysozyme, which breaks down, foreign bodies. So it's kind of like first line of defense. Anything that's open to the outside, like that's why your mouth, you've got tonsils guarding your throat, because you're getting stuff from the outside and putting them in. All right. So contains mostly water, of course, some salts. Have you ever tasted tears? What do they taste like? Salty. Yeah, just like sweat as well. It's salty. When you work out. So that's because of the salts in there. And of course the mucus. What will the mucus for in the tears? Lubrication, because that's the mucus. Lubrication. That's the lubricant part of it. Okay, and this is the sort of immune surveillance part of it, because it will break down, bacteria and so forth. So tears have a big function, right? So they enter the eye via the superior lateral, because the eye thing is on the top, superior and to the side. You will see a picture. So those superior lateral canaliculi will, or excretory ducts, because they are excreting, that's where the tears are coming from. Excreting ducts come down into the eyelids spread it around, and then medially via the lacrimal canaliculi. And also there's this little hole. I'll show you to you in a little bit. And then they drain into the nasolacrimal duct. So that's the sequence of the tears. So I can know the function of the tears, the components of the tears, and what those components are doing, and kind of like how the tears are channeled through the eye, right? So I need you to know that in terms of. So this is a one that shows you. So the lacrimal apparatus is on the top right here, okay, on the superior lateral part aspect of the eye. So the ducts kind of come in like that. The superior lateral ducts, excretory ducts, and then you see this is the little hole I was talking about. If you go like this, you will see it very easily, okay? And there's another one up here too, all right? Right there by the caruncle, by the medial angle of the eye, and then the lacrimal canaliculi take it into the nasolacrimal duct, all right? That's important. It's like an overflow, really. Otherwise, you know, if you didn't have that drainage into your nose, what would happen? Apart, you know, the meibomian gland is producing that sebum that's protecting the tears from running out. But if you didn't have an overflow, like into the nose, what would happen? You'd kind of always be crying all the time. Because, I mean, the tears are constantly produced and they are recycled like that. Through, so because otherwise it will get real dirty. If you're just recycling the same tears, they've gotten rid of bugs and whatnot. So the tears are that. So that's why you need that overflow. All right? So that's the movement of tears. So just know the movement of tears right there. Excretory ducts into the canaliculi in the middle, and then into the nasolacrimal duct. All right. Okay, so moving on with external structures, quite a lot of accessory organs that help out the eye. So this will be doing a little bit, in fact, the anatomy of it will be identifying, just now after we're done with the brain, we'll be identifying these extrinsic eye muscles, which there is 6 of them in the human eye. We get all 4 compass points with a muscle. The top one, superior rectus, the bottom, inferior rectus, the one towards the nose bridge, the medial rectus, and the lateral rectus on the outside. And on the top, we have an oblique muscle, that superior oblique that runs at an angle. You did? Wow. You enjoyed. And then, of course, the inferior oblique kind of runs from the lateral rectus. In fact, it's the way you determine which is the lateral and which is the medial, because the inferior oblique runs from it. All right? So they enabled the eye to literally to move the eyeball, to follow moving objects like this. And these muscles are controlled by the oculomotor. Remember the oculomotor, and the trochlea controls the lateral rectus. I mean the superior rectus, superior oblique. Okay, so and the oculomotor controls the lateral, the superior, and the inferior rectuses. All right, and also they maintain the shape of the eyeball too. Why do you think we must maintain the shape of the eyeball? Okay. And meaning that what? Mm-hmm. To the focal point, exactly. Yeah, everything becomes distorted. Why? Because there's a certain focal point, which we are going to call the macular lutea, where all the light is focused, where there's a concentration of all these photoreceptors, especially cones. Okay? So, if your eye is short, it's squished like that. Okay, let's say this is the ideal eyeball. And then your eye is squished. So that means when you focus light, you focus behind, because your eye is short. So maintaining the shape of the eyeball is kind of important. Okay? If your eye is long, because it's been squished like this from the top or whatever the effect is, then you're going to focus too early. See, here you focus too late. See, because your eye is too long.
Isn't that common? That's when you're starting to decide right?
It's not really that common. I mean, as you get older, your sight goes bad, though. But and we'll talk about something called presbyopia, where, you know, in older people they have to put the thing like far away like this to see, because their near point of vision goes further and further when you get older, because the lens cannot accommodate. When you're viewing near objects, your lens has to plump up like this. So as you get older, it fills with all this connective tissue and whatnot, and it becomes hard, and it doesn't accommodate. Oh, they can do lens replacements. They do that.
Having to do with astigmatism?
It's got to do with that and a whole bunch of other things, or even cataracts, to growing of connective tissue in the, in the eye, in the lens. So the lens cannot accommodate, and we'll talk about accommodation in a little bit. But these extrinsic eye muscles do that. They maintain the shape of the eyeball, and it's important. All right. 4 rectuses, of course, originate in the 4 compass points on the eyeball. All right, and then the 2 obliques on the top and bottom. All right. Okay. when we dissect through the sheep eye, it's a ruminant animal, and it has another, the seventh extrinsic eye muscle called the retractor bulbi, which we don't have. And that, no, because those cow eyes were fresh, and we tried to put them in that solution to preserve. They didn't last that long, and so I'm not going to use them again. So we're going to use real preserved. Hopefully we have a good set this time, because normally we get good eyes from them guys. So this is just looking from the top, and those are those extrinsic eye muscles. They kind of maintain the shape of the eyeball. All right. And we'll really visit them a lot in the lab when we are identifying the rectuses and so forth. But this just gives you a visual of how they are located within the eyeball. All right, and this is what they do based on their location. The inferior rectus on the bottom kind of pulls the eye like that, so it medially and deviates the gaze towards the middle and down. Sure. Is it this one you want? Yeah. So this is the inferior oblique muscle. And on this side, the one that's cut over here, that's the lateral rectus. All right? And you can see the superior oblique kind of comes at an angle like that and is hooked up onto this collagen tissue, and it goes like that. Goes towards the back. So it kind of pulls the eye to the middle and up. All right? This is kind of like how it occurs in the human, actually. It's kind of like an impression of a cadaver. That actually is the way it looks. Did you have anything specific that you...
No,
Yeah, I was thinking that, since we're going to actually cover them in the lab, we will pretty much go through them in greater detail. Because we get quite a bit of material to go through in terms of okay, no, but it's a nice picture though. It's really nice, and I'll post this, of course, as usual, so you can actually see these because you won't get these pictures anywhere else. And these are just the functions of them, and they're based on the you see, so the oculomotor mostly controls these, all right. And the abducens and the trochlea does the superior, and the abducens does the lateral, rectus. All right. Okay. I will cover mostly these ones, and I will be doing an electrophysiology experiment for these extrinsic eye muscles. I think you guys probably miss it. No, you are the guys who are going to do it. We will do, in our electrophysiology experiment, we're going to measure electrical activity as the eyeballs move. So when you move to the right, we're going to see a certain pattern of electrical activity when you move to the left, or when you look up, or when you look down. And then pretty much after the lab, you'll be able to see the electrical tracing and say, okay, this guy was looking to the right, or this guy was, you know what I'm saying? So we're going to do that, and we'll revisit these a little bit. I had to place them towards the end, and I couldn't line it up with the lecture because we need to get the equipment and all these computers, with the software that runs those equipment. All right, so that's the only reason why. Okay, so we're moving on to anatomy of the eye. It's a slightly irregular hollow sphere because of the cornea, right? It's mostly round like this, and then the cornea kind of bulges outside like that. So it will have the anterior part and the anterior cavity over here, and it's separated by the lens over here, and then the iris will create another cavity. So you have the anterior cavity, and then the space between the iris and the lens will be the posterior cavity. I know it's kind of strange. And then the one that's really at the back will be the vitreous cavity, because of the vitreous humor in there, okay? All right, so, and we'll talk about that in a little bit. All right, so these are the three tunics. The fibrous tunic will have the sclera, which is the white part of the eye. It's very tough. When you cut through it, you will see how tough it is. And then, of course, the cornea on the front. The cornea is very tough too, and transparent. And we'll talk about some of the characteristics of it that renders it colorless or transparent. Why does it have to be transparent, the cornea, for instance? To allow light to enter. Yeah. So we got those two. The vascular tunic is got all the blood vessels of the eye, and it's the choroid cord. It's got all the blood vessels of the eye. All right. And then the ciliary body and the iris. The ciliary body are muscles that radiate around the front part of the eye. All right. That will suspend the lens. And the ciliary body are attached to suspensory ligaments that are actually attached to it. So you've got the ciliary body, suspensory ligament, and then the lens like that. Suspensory ligaments, ciliary body. So it goes all the way around. So when we dissect through the eye, you'll be able to see that. And then, of course, the iris, which is the curtain of the eye, the one with the radial and the circular muscles. And we'll cover each of these separately, especially the iris, because it regulates entry of light into the eye. And then, of course, the nervous tunic will do quite a bit on that too, which is the retina, and includes the rods, which are active in dim light conditions, and cones, which are active in light. And therefore, cones are responsible for color vision. Cones. All right? So we'll do those receptors towards the end. All right? And then, of course, we've got the internal cavities, which are filled with the fluids. The anterior cavity is more aqueous fluid called aqueous humor. And then, of course, in the posterior cavity, right at the back, the vitreous cavity, we have the vitreous humor, which is a gel-like. And when we dissect through the eye, you can see if the eye hasn't been damaged, you can actually put the gel on your plate as if it's like a marble. It will be, yeah. Sometimes it's been damaged and it's watery, but that's what it is. Because what it does, the purpose of that vitreous humor is to hold the retina flat against the choroid cord. Because if the retina detaches from the choroid cord, then you can't see. And in fact, there's a condition called retinal detachment. Sometimes they actually take out the vitreous humor and infuse artificial humor in there, so that you can really hold, and it also maintains the shape of the eyeball too. Yeah, retinal detachment, like if you have a very traumatic injury to the eye, you could have that, but it also occurs as you get older. As you get older, yeah, it happens as you get older though. Yeah, but if you're young, it normally happens due to traumatic injury to the eye.
Isn't there like, I'm trying to blank on what it is, but isn't there some sort of genetic disease or something that they like test the pressure of your eye because of that?
Intraocular pressure?
Yeah.
Yeah, there is that too, because the fluids in the eye is also homeostatically regulated so that it stays the same, and it's kind of recycled as well, especially the anterior part of the eye, because the anterior part of the eye, the cornea doesn't have blood vessels for obvious reasons, right? If it had blood vessels, the cornea would not be transparent, you see. So, but it needs nutrients because it's a living tissue. So that that fluid is recycled, and something goes screwy, the pipe, well, I call the pipes, but they're ducts, right? But they get blocked, and or something is inflamed and it gets blocked, and then the fluid is accumulating in the eye, and it increases the intraocular pressure. So if you have a genetic condition where some of the proteins that are involved in there are not functioning properly, and you are not recycling properly, and you get accumulation of that fluid in there, the pressure goes up. And sometimes it could be really bad. If you go for your eye exam, they put something in your eye to measure ocular pressure, to make sure that it's quite correct. What's that? Yeah, sometimes with me, my thing is it's got a little kind of pin-like thing with a little ball at the end of it. It's like a needle, and then it just goes ping. It's very ticklish that I always can't keep my eyes open for it. So they just kind of do that. They put it right on there, and then it just shoots a little dart-like thingy. It's kind of scary, you know. The eye, you know, you'd think is very delicate, but it's the toughest thing on there. Once you cut through the cornea, you will respect the eye. It's tough. The cornea is this thick. Yeah, it's just in your brain. You think that that little pin is going to hurt you, but it won't, because the cornea is just so thick. It just couldn't do anything. Yeah, the cornea is. It's amazing how thick and tough the cornea is. You will see for yourself when you cut through it. Yeah, that's, what was she doing?
Running with scissors. I don't know.
What was she was going like this, I guess. Oh, sorry. You know.
So it's kind of funny because her pupil is like a different shape.
Yeah. Oh, my God.
Perfectly fine.
Yeah.
See out of it.
She can see out of that eye. Oh. Phew. Maybe the scar tissue.
No, she's like 60 now. She can wear contacts and she's fine.
Wow, it's amazing. It's tough. Yeah, the vitreous humor in it. Yeah. These are your favorite pictures, Jessica, right? You know. So we're going to spend a few seconds, you know what I'm saying? You know what I'm saying? There's this ganglion, though, I wanted to point it out. So you get the short neurons that come through here. You see this ganglion here on the optic nerve. So you have short neurons that come and synapse here, and then the synapse with the longer ones that will eventually go to the eye. Okay, so this is why it's showing the short neurons over here. But then the optic nerve over there is just cut. And the area where the optic nerve enters, the eye is the blind spot or the optic disc, because on that spot there are no light receptors. In fact, there's an exercise you can do to prove the existence of the blind spot, and I can show you at the end. But you know, you just look at some red dot and then a little plus sign. You look at them. Focus on the plus sign, but you'll be able to see the red dot. But and you put the object closer to you. As you put it closer to you, once it hits the blind spot, you won't be able to see the red dot anymore, and you just be able to see the plus sign. So because there are no photoreceptors there. So anyway, I like this, the way they set up this, because it's just like in a cadaver, you can actually see you know, when they dissect through. All right, and so that's that. And then of course the brain, which we are actually doing right now, and you know, the optic chiasma, which is the optic cranial nerve number 2, and then the optic tracts, and then some of the other cranial nerves which are not cut on this, because this is an actual human brain. And then of course you have the optic nerve, and then the tracts. Once they're in the brain, they're called tracts. Okay, optic tracts, which is over there, from the optic nerve, and then the tracts. All right. So, and there's the brain stem, and the mammillary bodies over here. Right, which is what we're doing right now in the lab. Right. Okay, so that's another cool picture there, showing you the eye with the vitreous humor at the back over there, and then of course the sclera outside, the choroid cord, and this yellow part. In fact, the retina, when you open the eye, it's kind of like a creamish substance. It's like a cream that's been smeared in the back of in the eye, and then of course the vitreous humor gel is actually holding it in place. So one of the things is that when you open the eye and you remove the vitreous humor, you have to look very quickly towards the back where light is focused, so you can see the macular lutea. It happens as a sort of because it's kind of fawn colored, almost like straw colored, the retina is. And then the spot where the light is focused, the macular lutea appears darkish, a little darker, fawn color, or sort of yellowish. No, I'm talking about, that's the choroid cord, which was greenish, blackish. Yeah, but the retina will be in front of it, and that's the one that was kind of straw colored, kind of fawn colored, yellow, light yellow, if you will. But the spot where the macular lutea is, where the concentration of the cones is, it's going to appear a little dark, but it disappears very quickly when you expose it to light. So sometimes you don't see it, and if the retina sags, because it's a creamish, it's almost like a cream, like cream cheese. Well, except it's a little bit more grainy, it looks grainy like. But most of the people don't see the macular lutea because it disappears real quick. So that's one of the things I wanted to point out. So this is the ciliary body right here with the suspensory ligaments. The ciliary body and this is a muscle that contracts and relaxes to accommodate, to change the shape of the lens. All right? Accommodation happens when you're viewing things less than 20 feet away. As you bring them closer to the eye, your lens becomes plumper and plumper and plumper. Okay? And we'll talk about how accommodation occurs too. All right? And then, of course, the clear transparent cornea on the front over there. All right? So we got quite a bit to go through. We got the fibrous and then the vascular and then also the nervous. So it's the posterior 4/5 of the eye, because the 1/5 is the cornea. All right? And it's opaque and is what appears white. And it's tough too. You will see when you cut through it, it's tough. It's like a little tennis ball. Have you ever cut through a tennis ball? Yeah, and the tennis ball material makes it stay like that too. There's a bit of air in it, but it's also the material that keeps it round. That's also the purpose of that sclera, to maintain the shape of the eyeball right there. You see what I'm saying? Because of how tough it is. And then provides the site for muscle attachment. Of course, those extrinsic eye muscles are attached on the cornea, on the sclera, on the sclera, okay? On the sclera. And then the cornea is the front part, which is transparent, and it's curved like that. That's why the eye is kind of like an eyeball, but it's kind of irregular a little bit because of that curvature on the cornea, and that curvature is meant to refract light. The more curved it is, the more, in fact, it's responsible for most of the refraction of the light. The cornea is. All right? Okay, so those are the two guys in the fibrous tunic, and this is towards the back, the sclera, and then, of course, the cornea, which is transparent towards the front. All right? And the cornea is so shaped, the convex shape kind of brings, focuses light together, comes together to converge, to come together. And then a concave one, which is the opposite of that, will kind of diverge light, it will spread it out. So then that, we'll talk a little bit more about that when we talk about correction of nearsightedness and farsightedness. All right? Towards the end. So we got the choroid cord, which is a vascular network, provides all the blood vessels. When you go to the ophthalmologist and they shine a light through your eye, they can see the network of those blood vessels. And I've got maybe a picture or two on this, presentation, but I may have removed some of them. But they will probably be one. A vascular network, that's why the choroid cord kind of appears black and dark because of the melanin in it. All right? The melanin in it, of course, you're getting light rays from the sun, okay? And that's what causes mutation. So you're going to need a whole bunch of melanin to protect you from that. That's why it's so black like that, because of the melanin content, okay? And also, when you go to the movies, during the day, they close the light out so you can actually see. Same thing, you don't want the light to come out. That's why I switched off the light here, because then you want to be able to see light coming from the projector so you can actually see those things. Same thing, the eye does that. It doesn't want light to be wasted and to go outside, so it contains all the light that has been gathered and focused. It has to focus onto the macular lutea so you can actually see it. So that's another purpose of that. All right. So if I ask about some of the purposes of these, accessory organs, you'll be able to kind of tell me, because most of these are kind of intuitive. They make sense. All the functions from the eyelids to the tears and all that. It makes sense. So you don't even need to really think hard. You just need to imagine yourself and what would be best for your eye function, and you can come up with pretty good reasons for it, for all these. And then, of course, the ciliary body is a ciliary ring that surrounds the lens and suspends it right at the junction of the vitreous cavity and the posterior cavity. all right, thickened ring of muscle surrounding the lens, and it's composed of smooth muscle bundles. This contract and relax. In fact, when the ciliary muscles are relaxed, it's flat, and therefore it will pull on the lens. You know what I'm saying? It's a very counterintuitive thing that it's hard to imagine. If a muscle is relaxed, it's flat like that, right? It's just like my biceps. If it's relaxed, if I go like this, they will get a peak. All right? So if it's contracted, it's going to pull on the lens. So the lens is more flat. See what I'm saying? If it's relaxed. So this is the ciliary muscle relaxed. So if the ciliary muscle is contracted, it becomes shorter and bulges out. So now it gives more slack to the lens, and the lens is more taut. I mean, it's more curved. Do you see that? Can you imagine that now? When the ciliary muscles are relaxed, the lens is taut because it will pull on it, because it will be flat like this. But when it contracts, you see how the muscle becomes short and it piles up on itself, and it creates all this distance here. So there's more slack on the lens, and the lens is more curved. Here, because it's flat, you only have this much give on it, so it's going to pull on the lens, and the lens is, you know. And we're going to talk about it later on when we talk about the emmetropic eye, which is the resting condition of the eye, when the lens doesn't have to accommodate or to plump up to view objects. So those will be objects at about 20 foot away. The ciliary muscles are relaxed. So that's like the natural, you know, shape of the lens. And it's called the emmetropic eye, and we'll cover that in a little bit. All right? So, and then it anchors the suspensory ligaments that are suspending the lens in the posterior cavity, right? And then, of course, the ciliary body, when it contracts and relaxes, changes the shape of the lens. All right? When it's more plump and more curved, it means it's going to bend light more. So that means you're viewing things that are near to you, because that's when you need to accommodate. When things are 20 foot away, your lens doesn't have to do anything. The ciliary muscles will relax. So that's the natural condition. It's called the emmetropic eye. All right? And then, of course, ciliary processes, which are kind of radiating outside, they also make aqueous humor, because it's right at the junction of the vitreous humor and the to the front. It will be the, you know, the posterior cavity and the anterior cavity. Aqueous humor circulates in there. All right? So that's the ciliary body. All right? So then you get the choroid cortex, the second layer after the sclera. All right? And then, of course, you get the ciliary body right here with the muscles and also with the suspensory ligaments. All right? So you can see when it contracts, it kind of gives, because the muscle piles up on itself. When it relaxes, it flattens out, and the lens is also taut and it's flat. It's one of those counterintuitive things that is the opposite happens. When the muscle is relaxed, the lens is taut. It pulls on the lens. Okay? So, so the vascular layer, okay, continuing. The iris now, which is the curtain of the eye, is got two muscles. Remember I said the radial muscles that radiate outside? Okay, so those are the dilator pupillae, because they dilate the pupil. They open it up. The saccular muscles are the constrictor muscles, sphincter pupillae, because they will constrict the pupil. Okay, so you can imagine the sphincter pupillae would be active in what conditions? Dim or light conditions? Pupillae, bright light. Because if you've got too much light coming in there, you squint your eyes like that. It will blind you. So the sphincter pupillae will be active in very bright light conditions. And then the dilator pupillae will be active in dim light condition, because then you really need to open up so you can let as much light as possible, because there's not a lot of it. Okay? So in the exam, I usually ask you to describe the function of these muscles in the iris and under which conditions they work. Okay? And why is it? All right? So close vision, when you accommodate, apart from making the lens very curved, you also constrict if you know, you can prove it yourself by bringing an object close to you. Your pupil will start to constrict, because then the light coming from that object is very close. So, and there's going to be a lot of it. So your eyelids and your eyes move medially like that. Have you noticed it? Bring your thing and you can do that. All right. So those three things happen: accommodation, and then of course the constriction, and then the eyes converge. And we'll talk about it in a little bit. Color is determined, of course, by the amount of melanin in there. So if you've got blue eyes, which happens in fair-skinned individuals, then there's not a lot of melanin in your eyes. All right. If you've got brown, like myself, or black, then there's a whole bunch of melanin in there. All right. And then light brown, green, and gray, and so forth. Those eyes, there's medium amount of melanin. All right. Okay. So you never really see, although there are some dark-skinned individuals that have blue eyes, because they have too much melanin in there. So that's just what happens. And the iris is one that's colored, right? All right. So yeah, that's what gives it color. The melanin. The more melanin you have, the more black it is. Just like in the choroid cord, that's why it appears, dark green to like black. The choroid cord does because of the melanin in there. All right. And the melanin is really supposed to protect you from the sun rays, because that's what the light source is, And these are the dilator pupillae and the sphincter pupillae muscles, radial, this one's on the outside, and circular or sphincter pupillae muscles. You can see you're in dim light conditions here, and you can see here you are in bright light. That's when you squint your eyes and you constrict your pupil, right? So this is the active muscle, that's the active muscle. And you can tell me why that is, right? When there's too much light, you want the smaller hole on there, on the pupil. When there's less light, you want a bigger hole on there. So you open it up, so you can get as much light in as possible. All right? There you go. And then the nervous tunic, this is the retina. That's where we have the rods and the cones. All right? 126 million photoreceptors in there. That's a lot. It's like amazing. The body never ceases to amaze. It's a gift that keeps on giving. That's a lot of photoreceptors in there that respond to light. And it's in the inner eyeball, and like I said, it's kind of smeared right at the back of it. And it's usually kind of fawn colored, as the back. So this is the point I was talking about. It's called the macular lutea. It's a bigger spot where all the light is focused, and it's concentrated with cones. Most of the rods are on the periphery. Why is that? You remember I told you what are the cones responsible for? What's that? Cones for bright, and therefore color vision. During the day you can see color, right? At night what happens? What do you see? You see the outlines only of the, and so those are the rods that work at night. Okay? So when you're going to have a light concentration on there, you're not going to have a whole bunch of rods because they work in the dark. So the rods are on the periphery, but the macular lutea has got a concentration of cones on there because the cones are the ones that are active in bright light conditions, and you are able to see color during the day. Okay? And then the center of the macular lutea is what is called the fovea centralis. That's where you have the sharpest visual acuity. It's the center of the center, if you will. Right? The center of the macular lutea is called the fovea centralis. So, and then you have the highest concentration of photoreceptors. And then, of course, the optic disc, like I was talking about, that's where the optic nerve gets into the eye, and that spot is devoid of photoreceptors. So it's not sensitive to light, and you can't see there. All right? So that's the blind spot, sometimes alternatively known as, because there are no photoreceptors. All right? Okay, good. So that's the optic nerve right there. And then, of course, the optic disc is right there. And then the retina, that's where you have all the photoreceptors. All right, and then the macular tear may be somewhere here or something like that. Is that spot where all the light is focused, all right. Yeah, this is the picture I was talking about. Oh, I do have one. All right, that shows you. You see, the macular tear is the bigger blue spot, and then the center of it is what's called the fovea centralis. That's the sharpest vision ever over there. And then, of course, the optic disc, and you can see from the optic disc where the optic nerve gets in. That's where all the things radiate, the blood vessels, right? Okay, so those are the retinal vessels. They have to spread nutrients throughout nicely, because if one is blocked or something, part of your retina is not going to get nutrients, and you may have problems and so forth and so forth. So that's why they always look in there. So these are the three chambers I was talking about. The anterior chamber is just behind the cornea, but in front of the iris. And then behind the iris, but in front of the lens, is the posterior cavity. All right? And then behind the lens would be the vitreous cavity, and the vitreous cavity contains that gel that holds, vitreous humor that holds, and it's really like a gel. It's almost like a marble. If you remove it and your eye hasn't been damaged, and it will sit on the table like a marble. And then the posterior, in the posterior and anterior, you circulate the aqueous humor, so-called because it's more watery. Okay. And so we'll talk about each of those. The chambers of the eye, aqueous humor. So this is what I was talking about, the function of aqueous humor. And the biggest function of aqueous humor is to provide nutrients to the cornea, because the cornea does not have blood vessels. It's devoid of blood vessels, because otherwise it will be red and you can't see through it. Yeah, true. Because hemoglobin is red and it's pigmented, so you couldn't. So fills the anterior and posterior chambers. The posterior chamber is one just behind the iris, but in front of the lens. All right, and then so supports, nourishes, removes waste. All right, and of the cornea, it does what the blood vessels do. When the blood vessels go there, they collect carbon dioxide, waste, and so forth, and deposit oxygen and things like that. So that's what the aqueous humor is, like the blood of the cornea. All right, because it doesn't have blood vessels, so that is transparent. All right, so produced by the ciliary processes. Remember, the ciliary processes make aqueous humor, so it's constantly made and constantly recycled, because it's doing the act of bringing in nutrients, taking out waste. So you've got to recycle it. So you don't want any blockages in that, otherwise your eye, will start getting infected and all sorts of problems. All right. And it's blood filtrate. Makes sense? The ciliary processes will filter blood. Almost, it's like a blood exudate that will have all the goodies in blood, minus the blood cells and all the hemoglobin with the red, and then it will supply that as a pretty much a transparent fluid, but it will contain all the nutrients, all the oxygen in the blood. And sometimes it may need to carry hormones in there or whatever dissolved substances for regulation and things like that. So it's retained through the scleral venous sinus. A sinus is a space where blood is collected back into the venous system. All right, like the dural venous sinus takes back some of that fluid, cerebrospinal fluid in the brain, and the same thing with this scleral venous sinus, because you've got to return it back into the blood and then recycle it again. You bring some new blood, and then you take the exudate out of it, or filtrate of the blood, and then supply again, and it goes back into the blood system again. So that's how it does. It's just a way to remove the color in the blood, but to remain with the nutrients. So that's what the aqueous humor does. All right. So vitreous humor, on the other hand, is also a colorless gel for obvious reasons, right? So that light will go through it, and also acts as a refraction as well. Also refracts light. All right. The main responsibility for refraction is the cornea, but the lens also refracts light. And the vitreous humor also does that. So filled vitreous chamber contributes to intraocular pressure. Remember intraocular pressure? Because what is important for is to hold the retina flat against the choroid coat. So it just has to have the right amount of pressure to do that. If it's too much, it's no good. If it's too little, then the retina will start to detach from the choroid coat, and then you have visual problems. Okay? And then, of course, it helps to maintain the shape of the eyeball, remember? Together with the extrinsic eye muscles. That's why it's gel-like. Holds the lens in place. You know, in fact, in fish, because the refractive index of water is a lot more than air, because, you know, water is denser, right? So fish, to see, they move their lens back and forth, actually. Yeah, because just the bending of it is not going to be enough, you see, to focus the light. So fish do that. They have a lens that moves back and forth to actually do that. Yeah, it's interesting. All right. And then, of course, it refracts light. All those reflects light. All those things reflect light. All right. And the lens is also has to be transparent. All right. It's made of lens fibers. All right. It's biconvex. It means it has a curved surface like that and a curved surface like that. So it's biconvex. All right. Okay. It's avascular for obvious reasons, right? No blood vessels in there, because then it will be opaque and then you can't see through it. It's transparent, it's flexible, and it's made up of lens fibers. Okay, okay, and it's got this colorless protein called crystalline, and it's arranged in a very regular fashion. All right. So to maintain the transparency of it. It's arranged in a very regular fashion, in an orderly way. Okay, you can imagine if the fibers are all over the place, it's not going to have. You could bend light in any one direction. You know what I'm saying? So the lens fibers have to be oriented strictly in straight lines or in a very orderly fashion, so that the refractory properties, the clarity of it. Because if things are kind of all over the place, it's not clear. It's murky. Yeah. That contributes to the clarity. That contributes to the refractive index of the lens. Okay. So all these things, you can talk about the characteristics and give reasons for it, or the functions behind that. All right. So it allows the precise focusing of light, because the cornea is also doing light refraction. And this kind of sort of finishes the job. The retina does. I mean, the lens does. All right. So this is a colorless protein. All right. So with age, what happens to the lens? It becomes tough because of the extra collagen that goes in there. It becomes dense. It loses the elasticity. So it won't be able to accommodate. So you can't look at things very near to you. You want to just view things that are 20 feet or more. So as you get older, it's called presbyopia, where as you get older, the near distance of sight is longer and longer and longer. So sometimes, yeah, you need reading glasses, so you could help to, focus the light. Sometimes old people say, if I had longer arms, I'll be able to read, because then I could hold it far away. You know, you kind of run and then you say, oh, now I can read. And instead, because your near vision goes, in an 8-year-old, it's about 60 inches. But in a 20-year-old or younger, it's about just a few inches. You know, about 4 or so inches. So with age, that happens just because the lens is no longer, flexible. It has lost its elasticity and all that. Okay, so electromagnetic properties of light, all right? They're all energy waves from the gamma rays all the way to the radio waves, which are long. The gamma rays are very short. And the visible spectrum is so small, you'll probably laugh. But that's where all the colors that we can see are. The rest of the spectrum we can't see. X-rays, infrared and whatnot, we can't see all that. So it's the portion of the electromagnetic spectrum that the eye can see. It's very short, very, very short. And I'll show you. So we know about refraction, what refraction is, bending of light. So when it strikes a concave surface is like this. It's like a thingy, a cave. So it's a concave. It will diverge light out. When light comes through like that, it's diverged out like that. All right? And then when a light strikes a convex lens, on the other hand, like such as the lens, it will come in and kind of sort of follows the outline of the thing. So, and then that's converging. And then that's diverging. All right? And therefore, if you are farsightedness, you use a convex lens because you are focusing behind, you bring it closer. If you are nearsighted, you're focusing in front of the retina, then you need to diverge the light out so that it kind of focuses later. Do you see that?
I have a question.
Yes.
You know how infants can't see very far away? I mean, essentially they can only see a few feet away from themselves. Why is that? What part of their eye is not fully developed? Is it their lens?
The lens, when you look at far objects, we'll talk about the distant objects in a little bit. The lens that's when it's relaxed. It's not bent. It's not anything. But at the same time, the kids will see things at very close by. They will have visual acuity. But the far, it's just development of the light entering properties, because if you are looking at an object very far away, you've got to gather all that light and concentrate it into your eye. So I guess it's the immature eye, it can't do that efficiently yet because, one, it hasn't grown to full size, for instance, right? Because a kid is still developing, their eyes are still smaller, so they can't do it as effectively as an adult who's not old. Yeah. Yeah. It's got to be that. Yeah, and they're building up their cones as well, because when you're growing, we have 126 million photoreceptors. The child will probably not have 126 by that time. You know what I'm saying? They will have less, so they won't be able to really see very well. Okay, so and it gets better as they grow and so forth. So those are the convex and that's the concave lenses and so forth. So and I'll show you a picture of correction of near and farsightedness. Do you see what I was saying? This is this little speck of the spectrum that we can see. Just that. Okay? You see how wide it is? So maybe there are other creatures in the universe that see some of these. They could see X-rays, or they could see radio waves, the long ones or whatever, or microwaves. Who knows? I mean, if the universe is as big as it is, as they say it is, and we're like, Earth is like a grain of sand, there's got to be something else. You know what I'm saying? Why would all that be? If it's true, what do you think? If it's true, I think there's got to be some other, creatures that can see some of this, because it's such a huge, and there may be a purpose for some of that. And we use that, microwaves to cook and so forth. X-rays, you can see inside. Because if you had X-ray vision, I could see inside your organs right now. That would be freaky. So maybe that's why we don't see the whole spectrum. You know what I'm saying? Yeah, yeah.
Sensory overload.
Yeah, sensory overload. Yeah, it will freak the crap out of you. Seriously. It will. It's psychological too, there you go. So the functions of the complete eye, the cornea, responsible for most of the converging of the light. The aqueous humor, we talked about many functions of it, applying nutrients, supplying nutrients, taking waste from the cornea, the aqueous humor does. And then, of course, the lens, finally adjusts, the focusing of light and accommodates, which is vision closer than 20 feet. And then, of course, the vitreous humor, of course, does that, intraocular pressure, holds the retina front against the choroid cord. All right, and also refracts light. All these are kind of aimed at focusing and bending light to that focal point, right there to the macular lateral. Okay, so this is what I was talking about. We're going to run out of time. Anyway, so this is the distant vision thing that you were talking about a little bit there, Jessica. But distant vision, when you're looking at objects that are 20 feet and further, all right? And near vision, when you're looking at things that are closer than 20 feet. that's when you have to accommodate. When you've got 20/20 vision, you can see at 20 feet what a normal human can see at 20 feet. But they don't always equal good vision, you know. But, it just means you can see at that distance, okay? So that's near, so relaxation of the ciliary muscles, which we were talking about. When things are 20 feet away, your lens does not have to accommodate. It doesn't have to do anything, and your ciliary muscles are relaxed. And that's the emmetropic eye, which is the natural condition of the eye. Okay, when it's not, the ciliary muscles are not doing anything. But in that case, the lens is actually taut. It's more flat. Okay? So that's the normal resting condition of the eye, the emmetropic eye. All right? Far point of vision, the lens does not have to thicken, which is 20 foot for the average human being, right? But when it's near, that's when you have to start accommodating. That's when you have to start constricting your eyes. That's when you have to start, increasing the curvature on your lens and so forth. All right? And near point of vision, we're going to talk about it on this. All right? So you can see here, far point of vision, the suspensory ligaments here, there's more tension because the muscle is flat. And here, because the muscle is contracted and it is piled up on itself, there is less tension. It's counterintuitive, but that's what it is. When you're viewing things that are near to you, and the lens is more, plump, and it's more curved. So myopia, you are short-sighted. Being myopic is being short-sighted. You see, you're focusing in front of the retina. All right, so you need something that's going to diverge the light out, like a concave lens, so that you focus later. You see what I'm saying? And you focus on the, there you go. So that's why farsightedness, you're focusing it behind, because then again, they also need to maintain the shape of the eyeball. If your eyeball is too short, you'll focus behind. If its eyeball is too long, you'll focus in front. So maintaining the shape is very important. So you need a convex lens so that you can bring the light closer sooner. Okay? So if I ask you to explain what fixes farsightedness and nearsightedness and which lens you use and why, you'll be able to do that, right? Yeah, you should be able to do that. You can see this later on. Let's see how far we can go. Okay, we'll try to push the envelope a little bit so that we can do as much as we can. So when objects are less than 20 feet, that's when you need accommodation. There are three things that need to happen. We've been talking about it all this time. One of them is the accommodation, right, which is making the lens more curved so that it bends light more. And the other one is constricting the eye so that, because the light source is much nearer. It's like bringing the sun closer to you. You have to squint your eyes, right? And then so you got to accommodate, you got to constrict, and then you got to converge. Bring your eyes together, like when you're and you can prove it to yourself too. When you're bringing your thing closer, your eyes are doing this. In fact, we're gonna see what kind of electrical activity happens when you're bringing a thing closer to you and your eyes are converging. That's gonna be fun. All right. So if I ask about what three things happen to view things that are less than 20 feet, you talk about the accommodation, converging, and also constriction of the eyeballs, right? So this one actually is kind of fun and it makes sense. I like things that make sense because they're easier to remember. Some things you have to use your brain to kind of imagine it out, and not with the eye, because everything kind of makes sense. All right. So of course the pigmentation comes from the melanin to protect against the UV rays in the sun. Okay, from the sunlight. So it also that's where you have your visual acuity, because light is focused on the retina. Okay, so the rods and the cones for color vision, CC, rods for dim vision. All right, and then there are bipolar cells that kind of modify and accentuate the signal before it's propagated along the optic nerve. All right. There are other cells too, like ganglion cells, amacrine cells, that are connected to the sensory nerves in the optic nerve, and these also modify the signal, or make it more optimal, if you will, make the signal optimal. So there's many of them, like horizontal cells here. They kind of have horizontal fibers, and so forth and dendrites and whatnot. And then, of course, the ganglion cells. The ganglion cells are the ones that connect to the nerve fibers. They form the nerve fibers of the optic nerve. All right? Okay, so we'll concentrate mostly on these two. The cones, so-called because they actually look like cones. The rods, so-called because they actually look like rods. Okay? All right? Go figure. There's nobody coming, so we will try to push it a little bit, guys, so that we don't have too much to do next time. All right? I hope you don't mind. So this is why I was trying to kind of get a move on. So the rods are responsible for dim light conditions, and the container, but you will see a rod has got discs with membrane in it that contain the protein rhodopsin, which is in the rods that's responsible for bright light, for dim light conditions. Okay, so rhodopsin is that pigment in there, and it's located in those and the discs are to increase the membrane surface area. So a purple pigment consists of opsin, protein opsin, which is covalently linked to retinal, which is sensitive to light. Okay, retinal is a product of vitamin A. Okay, that's why I say if you eat your carrots and some of those things, vitamin A containing things, you get good eyesight because of the retinal. All right. The retinal is one that switches on and off, the rhodopsin. Okay? Because you see, these are the discs that increase the surface area, and that's where you have this protein here. So opsin is the protein. It's a transmembrane protein. You see, this is the membrane, and this is the retinal. When light, impacts the retinal, it changes shape and then dissociates from opsin, so that you kill rhodopsin in light conditions. You see what I'm saying? You kill it because it's not sensitive in dim, in light conditions. Okay? So when it's dim and there's no light, so it goes back to the original shape, joins and opsin to form rhodopsin again, and then rhodopsin you can see in the dark. So that's why this is switched on and off by light. Do you see that? It's kind of sort of vice versa, but it works like that. When light is on, you switch it off. You activate retinal because it's sensitive to light. And then when you activate retinal, you switch off rhodopsin because it will dissociate from opsin. Because rhodopsin is opsin and retinal. All right. So exposure to light activates it, like I was saying, so that the retinal changes shape and dissociates from opsin, and you no longer have rhodopsin. So in light conditions, the cones are active, and the cones have three different, pigments in it for red, green, and blue light. All right. Okay. So this is one of the cycles I need you to know. When light shines on rhodopsin, it will activate retinal. That's what happens. And then retinal changes shape, and then it dissociates from the opsin. And then it's not active. And then in the dark, when there's no light, it converts back to the original shape, retinal, and then again associates with that opsin. And then you have rhodopsin, and you see in the dark. That's why you have a lag period. Like if you watch a movie during the day, you come out, what happens? You can't see in the light because you've got a whole bunch of rhodopsin in there, and you'll be killing it once the light starts to hit on the retinal, and then you kill it up, and then your cones become active. If you go into a dim from the light, because you don't have any rhodopsin, you can't really see. Afterwards, in the dark, it looks like you can really see properly. Have you noticed that? Yeah, that's what happens. It's because of this cycle. Because you have to make that protein and so forth. Okay, so know this cycle. Okay, and it's kind of like very intuitive, like I was saying. We're pushing it, right? We're pushing it because nobody's coming here. It's a Friday. You gotta pee. Oh my God. Cones. So cones do the you know, the color vision, and the vision of other colors in the spectrum is a mixture of you know, activating green cones, red cones, you know, and blue cones, and you can see all the visuals because these are like the primary colors. You know, back in the day when they first had the color TV, it had those colors on there, because when you mix any of those colors, you get all the other colors. All right. So this one is called Chlorolab sometimes. Okay, that's the name of it. This is Cyanolab, and the red is Erythrolab. Erythro is red, right? Okay, and that's in the cones, and then you can perceive other colors by mixing a different ratio of those colors, right? All right. And then what happens is that there's two types of red pigments. 60% of the people have the serine, because those are pigments, those are proteins. They have a serine, amino acid serine at position 180. 60% of the people. So they see a slightly different red. All right, and then some 40% of the people have alanine on position 180, and this is the spectrum you see. So that's why people have different color preferences, like your favorite color is different, because we literally don't all see the same red. Because, but we all see the same green and the same thing, blue. Mm-hmm. Yeah.
Yeah, but if you're red green deficient, that's
Some people are colorblind. Yeah, they don't see green. Or they, yeah.
just men that are colorblind.
Mostly men, because it's a sex-linked trait. It's on the X chromosome.
But that would affect your purple vision.
It would affect your purple vision because boom, boom, boom. So if you see somebody who's colorblind, who's green colorblind, the purple won't be their favorite color because they can't see it. See what I'm saying? Yeah, that's what happens. You see, because of the spectrum. So the one thing I wanted to...
As far as seeing red, and which red pigment you had,
Uh,
that affect your purple.
the purple is on this kind of side. You see the purple? Do you see it?
Right, that's what I'm doing.
Yeah. So yeah, and you can see some purple hues on this blue side. You may see some purple hues on this side, but they are more apparent on this side. Yeah. Yeah. Anyway, but that's the deal, though. We definitely see different reds. If you go get your thing checked, your pigment checked, and you see which one you have, so you know which wavelengths. Yeah, they could they can map your gene for it. They can map your gene for it if they take your cells, like your epithelial cells. They can your genetic, and then they know the locus for this gene, for the pigment, because they're all genes in there for each protein, and then they can sequence it, and they'll find which protein, which amino acid is on 180. They can do that. All right. You gotta pee. Hold it. So the fovea centralis, we have covered this, where the concentration of the cones are, and the center of the macular lutea, and then the rods, of course, I said, are on the periphery. So I covered this already when we were talking. Bipolar and ganglion cells modify the signal, or accentuate it, or to make it better. So we've kind of covered all these concepts as we were talking about this already, okay? And then, of course, there are interneurons that layers of that also enhance neurons between those ganglion cells and the nerve cells that pick up the signals, the receptors. All right. So all that is geared to modify the signal before it goes to the eye. You know that you see things inverted, upside down, and left to right. And then when it goes to the visual cortex up in the occipital lobe, then you see it the right way when you do perception. All right? So neuronal pathways of vision, of course, the ganglion cells are the ones that extend into the nerve, the optic nerve. And then remember, when they go into the brain after the optic chiasma, they become tracts. Any nerves, fibers in the brain are called tracts. That's it. All right? Just like the nuclei, parts, cell bodies in the nucleus, in the brain, and ganglion are outside. Cell bodies outside of the central nervous system. All right. That's why we call those ganglion cells that are synapsing at the optic nerve. Okay. So depth perception is the ability to see distance, and it's because of binocular vision. You know, when you're looking at an object, part of it is seen only by the left eye. Part of it is seen only by the right eye because of the light rays. But there's most of the a part of that object that both eyes can see. That's the binocular vision. That's the one that gives depth of the object and distance away from it, so that I can pick this like that. I don't overshoot it or pick behind it. Okay, it's binocular vision that is responsible for that. All right, and it's when eyes see the same light rays are seeing that part of the object. Okay, it gives depth perception and distance away from the person viewing. I wanted to just include this so that it shows the underside of the human brain, and you can see the optic chiasma and so forth. All right, this is a section that goes across. We're going to do a sagittal section. All right. All right. So this is what I was talking about. You see your eye. This is the part of the object that both of your eyes are seeing. Binocular vision. This is monocular vision because only one eye is seeing it. And monocular vision. This is what gives depth. And what do you call it? Distance. So you can see the distance of the object away from you. And binocular vision is that. Sorry guys, we went over, but we finished. I appreciate it. I'll give you 5 minutes, and then we can start the lab. Okay? Thank you guys. Thank you so much.